Hello and welcome to your first lecture in genetics and biotechnology. I am excited to be talking to you today about cellular respiration. So this lecture is a bit of a doozy. I'm not going to lie to you. It is got a lot of the inner workings of cellular respiration, which can become pretty complicated. I'm going to do my best to make it as clear and concise as possible, but I do expect you to know most of the inner workings of aerobic respiration, aka respiration using oxygen. So let's get into it. This lecture might be one of your longer ones this year, but I think you guys will survive. So let's start with some just like super basics. You know that living cells require energy, and energy comes from outside sources. Plants get their energy from the sun, and animals can either feed on plants, like the panda, right? Eat plants, or feed on other organisms that eat plants. So if you are not eating plants, you need to eat more because you get less energy from secondary consumers, even primary consumers. You don't get as much as if you were getting energy directly from the sun. Because we're talking about cellular respiration, we're not even going to talk about plants anymore. This is all about animals and things that eat other organisms to survive. Now, energy flows as sunlight and leaves as heat. That's kind of just like your basic understanding of how energy flows through any sort of ecosystem. It all starts with light energy, goes into a chloroplast, goes through photosynthesis, and whatever is generated from photosynthesis, right, oxygen and glucose, that goes into the mitochondria. We have mitochondria. We do not have chloroplasts, which is why we only care about, right now, we only care about cellular respiration. So when those organic molecules go into the mitochondria and cellular respiration starts to happen, you can use that organic molecules to generate ATP. Once you have ATP, that is the energy that can power almost all of your cellular work, and all of whatever is left over is released as heat. Releasing energy as heat is a waste. Okay, it doesn't do anybody any good to release energy as heat. So ideally, you want to be able to keep as much energy as possible so that you can do as much cellular work as possible. So you guys know a little bit about ATP, but let's make sure that you actually know what I need you to know for ATP. So it is your energy molecule. It's a power source for cells. I know what you're thinking. You're probably, as soon as I said mitochondria, you probably in your head said, oh, it's the powerhouse of the cell. Yeah, I get it. It is. But we're going to go way deeper, and that is not going to be an acceptable answer for any question I ask you. So ATP is your energy source. It has 3 bonded phosphates. In those bonds is where potential energy is stored. And then phosphorylation, which is just a really fancy word to say that a phosphate has been added to an organic compound. So when you add a phosphate onto ADP, adenosine diphosphate, right? When there's only 2 phosphates, when you add that 3rd phosphate in to make ATP, you actually make it much less stable. By making it less stable, it means it's really reactive, and those high energy bonds want to kind of break and release that energy. You can also add phosphates onto like glucose, which is actually what we're going to talk about in a little bit, but when you take these energy bonds and move these phosphates around and go through phosphorylation, you make molecules less stable. By making them less stable, you make them more reactive. And that's kind of what we're looking for. So through this, you have 2 key functions of ATP, right? Energy currency, which is probably what you already know ATP as. But what we're going to focus on is the transfer of that phosphate group to other organic molecules. So this is the idea of taking that 3rd phosphate and moving it from ATP onto glucose. And then we've talked about this before, right? Photosynthesis, cellular respiration, that's how you get ATP. That's like basic stuff. Hopefully you know that. So, redox reactions. If you are anything like me, I didn't particularly like chemistry. And that's fine. You can say that. I'm not going to tell your chemistry teachers. But redox reactions was one of those things that I learned about and then I completely forgot about because I thought I would never need it. And then here we are talking about redox reactions in a life science class. So, redox reactions are these reactions that either gain electrons or lose electrons. They're pretty important in this reaction and we're going to talk about them a lot, but what I want to talk about in this slide in particular is the idea of direct combustion or stepwise oxidation. So when you do direct combustion, which is basically this first picture right here, direct combustion, you have a really big activation energy, like you're burning it or something. And so the reactants have this huge activation energy and then an uncontrolled energy release, which means most of your energy has been lost as heat, which is, if you remember, I said that's a total waste of energy. So when you do a direct combustion, it's just totally uncontrolled, wild. You lose like the majority of your energy. It's just not worth it. If you're going to go through that, you'd have to be going through it all the time to make up for all this lost energy. But the way that we do it in aerobic respiration is this staggered stepwise oxidation. And by doing that, you have a much smaller activation energy and you do it way more frequently. And by doing that, you actually control the release of energy. And by controlling that release, all of the energy that is released is then sent to carrier molecules, and those carrier molecules can then use that further down the line in the steps of cellular respiration. Ideal, right? You want to keep as much energy as possible so you can use it all. So I want you to know the difference, but I want you to understand that cellular respiration uses these steps and not this huge, like, kind of blowout of energy and then just like uncontrolled release. It is a very controlled breakdown of organic molecules. So I gave you the definitions of reduction and oxidation here. If you remember, Leo the lion goes grr. I don't know if that was something that was taught to you, but I assume it was because it's pretty common. But Leo, L-E-O, loss of electrons is oxidation, so LEO, loss, electrons, oxidation. And then GER, gain, electrons, reduction, so GER, Leo, the lion, goes GER. If that's familiar to you, great. If you've never heard that before, hopefully that helps remember the difference between the two. Okay, so moving a little bit further into redox reactions, I talked a little bit about how carrier molecules gain protons or electrons, and then they can take them and transport them to be used further down the line. Well, in cellular respiration, we're going to just talk about NAD plus and FAD, which are two hydrogen carriers, electron carriers, however you want to call them, that are really common in cellular respiration. Most common is NAD plus, but we're going to talk about both. You don't need to know anything more about them except that NAD plus is reduced to form NADH and FAD is reduced to form FADH2. I mentioned this before, I'll mention it again. Hydrogen carriers transport electrons in cellular respiration, transport electrons to the cristae of the mitochondria, which is the inner folds of the mitochondria, but ideally what you know is that they transport electrons to be used further down the line in cellular respiration. Okay, aerobic respiration. This is like what we've been talking about this whole time. Respiration is the consumption of organic molecules and oxygen. We're going to focus on the consumption of glucose and oxygen. When you consume those, you get ATP. So you put in glucose and oxygen, you get out ATP. While the broad definition of cellular respiration does include both aerobic and anaerobic, typically when you say cellular respiration, you're referring to aerobic respiration, aka with oxygen. So again, for the purposes of what we are talking about in the next couple of slides, cellular respiration is going to refer to aerobic respiration. Cellular respiration refers to respiration with oxygen. Now you can use cellular respiration and your starting point, your consumption of organic molecules can be anything. It can be carbs, fats, proteins. Truly, it can be used by anything. But again, we are going to kind of strip away all of that and we're going to focus on one thing. We are going to focus on glucose. So that formula at the bottom is like your basic formula that I know you've all seen in freshman year biology. But if you are unfamiliar with it, your first bit here, glucose plus oxygen, yields carbon dioxide and water and energy, ATP. So you put in glucose and oxygen, you get out energy. It's kind of the beauty of this. Okay, I'm not going to go too deep into this, but I just want to kind of make sure that you are aware that the mitochondria is got kind of a lot going on inside. You've got the membranes, smooth outer, then an inner membrane folded into cristae, and then the inner membrane has two parts. But really, you should be aware that all of this is happening, but in the mitochondria, the areas that actually hold stuff that we care about is going to be the inner membrane space and the cristae. So as long as you understand that the cristae is a highly folded area, that is where the electron transport chain happens, I'm cool. Okay, so it's been a little bit of time, and we were just going kind of over the basics of the beginning of getting into cellular respiration. This is a bit of a long lecture. They're not normally going to be this long, I promise, but we're about to get into the nitty-gritty of cellular respiration. So buckle your seatbelts because it's about to get wild. So cellular respiration has 3 stages. Well, sort of like 3.5 stages, but 3 well-known named stages. So glycolysis, the Krebs cycle, or if you're like really fancy, the citric acid cycle. I am neither fancy or really fancy, honestly. So I'm going to call it the Krebs cycle, but you could call it either. And then the electron transport chain. This chain is also called oxidative phosphorylation, which is wild. I'm never going to call it that. So electron transport chain or ETC, they all mean the same thing. So I'm going to dive into glycolysis right now. Okay, glycolysis, step 1. This happens in the cytosol of the cell, aka the cytoplasm. Like flashing lights, big hint here, glycolysis does not happen in the mitochondria. It's key here, okay? It happens outside of the mitochondria in the cytoplasm of the cell. There's 4 key events that happen in this. Sorry if you can't see the very last bit of the slide, but you have phosphorylation. Remember, we've talked about phosphorylation as that addition of a phosphate to an organic compound, whatever that may be. You do this because through the addition of ATP. So glucose is phosphorylated by 2 ATP molecules, which means you need to start with ATP molecules to even go through glycolysis. So once glucose is phosphorylated, right, those ATP molecules have that extra phosphate which transfers to the glucose. That glucose is now less stable and more reactive. Once that happens, the lysis, or the breakdown of glucose, which is a 6C sugar, so glucose is split into 2 3C sugars. So it went from 6 carbons to 2 sets of 3 carbons. So it just like split in half. And then oxidation happened, which is our first intro to our electron carriers. So those hydrogen atoms are removed and NAD plus is reduced to NADH. Because there are two 3C sugars, you get two NADH molecules, one from each of the 3C sugars. And then ATP formation. Some ATP is produced during glycolysis, not a lot, but some of it. There we go, now you can see the bottom. So some is released. You end up with 4 molecules of ATP, 2 per 3C sugar, right? So 4 molecules total, 2 per 3C sugar. Now, remember that you needed 2 ATP molecules to start this. So even though you make 4, you really only have a net gain of 2. Not super helpful when you're trying to make a whole bunch of energy. Now, here we go. This is glycolysis in image form. Hopefully this helps explain what I was just talking about. So you have glucose, that's your starting compound. You introduce 2 ATP and you go through that phosphorylation. Once that's done, glucose breaks into 2 3C sugars. In here, you release some hydrogen ions, which allows NAD plus to turn into NADH, right? You get 1 NADH per 3 carbon sugar. And then as it goes through, you end up with 2 ATP per 3 carbon sugar. So your net gain, because remember you started with 2 ATP, so you kind of have to X out one of these gains here, is 2 ATP, 2 NADHs, and your end result is you have 2 pyruvates that you're going to use to enter the next portion here. Now, before we even get into the next portion, that was the first step of cellular respiration. I want to talk about a quick preview of anaerobic versus aerobic respiration. Both anaerobic and aerobic respiration go through glycolysis. The only way that we can move on to the rest of cellular respiration is if oxygen is present. So if oxygen is present, that pyruvate transfers into the mitochondria, it goes through the next few stages, and you get a ton more ATP. If oxygen is not present, pyruvate doesn't get broken down further, no additional ATP is produced, and the pyruvate remains in the cytosol. It can be converted into lactic acid or ethanol through fermentation, but it can always go back into pyruvate once oxygen is now present, and then that pyruvate can go through aerobic respiration. So we care about anaerobic respiration, but we care more at this moment in time about aerobic respiration. So this is our half step. The link reaction is what brings the pyruvate from the cytosol into the mitochondria. Really, there's not a whole lot to talk about here. We just want to talk about how the link reaction basically starts the actual aerobic processes of mitochondria. So the pyruvate is transferred into the mitochondria and loses a carbon atom. When it loses a carbon atom, it becomes a 2C, right? 2C compound, and that extra carbon forms CO2, carbon dioxide. That 2C compound forms what's called an acetyl group when it loses a hydrogen atom, and notice how you have an NAD turning into an NADH in the image below. And then that acetyl compound that you've kind of created combines with an enzyme, coenzyme A, and you end up with what's called acetyl coenzyme A or acetyl-CoA. It's a 2C compound, 2-carbon compound, and that's what you need to start the next step. So per glucose molecule, you end up with 2 acetyl-CoAs, 2 NADHs, and 2 CO2s. You're probably looking at this picture and saying, but Ms. Kalfa, I only see one of all of this. Remember that per glucose, which is what we started with in glycolysis, you end up with 2 pyruvates. So all of this is times 2. Now, the Krebs cycle, aka the citric acid cycle. I am not fancy enough to call it that. So this occurs in the matrix of the mitochondria. So now we're in the mitochondria. Acetyl-CoA, which is what we just ended up with in the link reaction, transfers its acetyl group to a 4-carbon compound called oxaloacetate. That's a real fancy word, and I'm not going to ask you about it, but I wanted to be as thorough as possible because this is a complicated thing, what we're talking about, and any extra information is probably going to help you make sense of all of these terms. So acetyl-CoA, which is a 2C compound, transfers its acetyl group to a 4C compound to make a 6C compound, 6-carbon compound called citrate. Now, do you see why it's called the citric acid cycle? So basically, a ton of reactions occur. I'm not going to make you memorize them all. When you get to college and you take Bio 101, whatever the intro to bio is, you probably will have to memorize them all. But right now, you just need to know that reactions occur that break citrate, that 6C compound, down to reform the original 4C compound, oxaloacetate. So there's a whole cycle here, and you go through the cycle twice because you have 2 acetyl-CoAs that start this, right? So you go through this whole cycle twice just to go from oxaloacetate to citrate back to oxaloacetate. I mean, I get it, but I don't get it. You know, it's just wild that the body does so many things. So I'm not really going to talk about all of the steps because, like I said, you don't need to know this, but you absolutely need to know the outcome. So down at the bottom, per glucose molecule, so after the Krebs cycle has happened twice, you end up with 4 carbon dioxides, 2 ATP, 6 NADHs, and 2 FADH2s. So now we've introduced both of our electron carriers. So this is the Krebs cycle. All of these reactions that happen as you go all the way around all of this, yeah, you don't need to know all of that. You need to know the outcome and that acetyl-CoA goes in to it, okay? So we're on our final stage, the third and final stage. We've almost done it. So I'm going to break this down into 3 kind of like mini parts. It all happens in like, it's all the electron transport chain, but quite a bit happens. So I'm going to break it down into like 3 bite-sized pieces. So this all happens in the cristae, which is that folded up inner membrane in the mitochondria. So this is like when you were drawing mitochondria when you were in 9th grade and you drew that like squiggle in the middle of a mitochondria of like the bean shape, that's cristae. So step 1 is generating a proton motive force. So what happens is NADH and FADH2 are oxidized. When they're oxidized, they release both an electron and a proton or hydrogen ion. Those electrons end up going into this electron transport chain, which is like this purple thing. When they go into the electron transport chain, they are passed through all of these proteins and they lose energy as they are going through these proteins. But those proteins pick up that energy, and those proteins then use that energy to pump these protons or these hydrogen ions from the matrix into the inner membrane space of the mitochondria. So NADH and FADH2 are oxidized, release an electron and a hydrogen ion. When the electron goes through, it gives its energy to these proteins. When the proteins have this energy from this electron, they pull and pump the hydrogen ions through from the matrix to the inner membrane space. So you go from you have a low concentration outside and a high concentration inside. Now, we care about this because it creates a gradient or a proton motive force. That's step 1. Step 1 is literally just what I explained. Pushing the electron through and making this gradient. Now, step 2 is now that we have all this, you've kind of got your de-energized electron here, right? But now you have these hydrogen ions up on the inner membrane space, and they're in this high concentration. But I know you guys know about osmosis. They don't like to be out of balance. They like to be equal. So they want to go back to the matrix for balance. And this diffusion is called chemiosmosis. Okay? That's a key word, chemiosmosis, because chemi means like chemicals, right? There's like enzymes involved. And then osmosis is the diffusion of this through this enzyme here. So the way that chemiosmosis happens is it pushes these hydrogen ions through this enzyme called ATP synthase. If you know anything about those terms, even synthesis, you can probably guess what's about to happen. So as the hydrogen ions pass through this ATP synthase, they trigger this enzyme to rotate, and by rotating it, it actually produces ATP. So because those hydrogen ions move through it, you get ATP being synthesized. That's step 2. Step 3 is kind of like the last bit, the cleanup crew, if you will. So step 3 is the reduction of oxygen. So the electron transport chain cannot continue with all of these de-energized electrons and all of these hydrogen ions just hanging around, right? It's like trash that needs to be taken out. So oxygen acts as the final electron acceptor. So once this electron travels all the way through the electron transport chain, oxygen shows up and is like, hey buddy, you want to ride? You seem pretty beat up. And so the electron and those free protons that have just gone through ATP synthase hook a ride with oxygen, and it turns that oxygen into water. So now you've maintained the hydrogen gradient by not adding additional hydrogens into the matrix area, and you've created water, which is always helpful. So if oxygen is not able to show up at this point, ATP production is halted. You can't do anything else because now you just have a buildup of like trash, basically. Okay, so you need oxygen to show up to clean up after everybody else. Okay. We did it, sort of. We got a few more slides, but we pretty much did it. So this is just a summary. So I'm just going to go through it really quick. You always, for our purposes, start with glucose. Glucose goes into the process of glycolysis, and you end up with 2 ATP, 2 NADHs, and 2 pyruvates. You use pyruvate to go into the link reaction. The outcome of this is 2 CO2s, 2 acetyl-CoAs, and 2 more NADHs. Acetyl-CoA goes into the Krebs cycle, and we go through the Krebs cycle twice. The outcome of this is 2 ATP, 4 CO2, 6 NADHs, and 2 FADH2s. All of these electron transport carriers go into the electron transport chain along with some oxygens. After the electron transport chain, you end up with 32 ATP and 6 molecules of water. That is your total outcome. Now, we talked a lot about what happens with oxygen, and we're just going to do a very brief overview of what happens without oxygen. So glycolysis can still happen, and if oxygen is not present, glycolysis will go into fermentation or anaerobic respiration and produce a minimal amount of ATP. Although anaerobic respiration still has an electron transport chain, it doesn't use oxygen. Obviously, fermentation doesn't have oxygen, so it can't use oxygen, but it still has an electron transport chain. So the final electron acceptor in fermentation could be something like sulfate. Again, I want you to learn one ETC, and that's going to be for aerobic respiration. So fermentation also uses phosphorylation, but it uses phosphorylation to generate ATP. So instead of using that electron transport chain and all of those steps, it just uses phosphorylation, which doesn't produce a ton of ATP, which is why fermentation is not ideal for like humans. Okay. So fermentation has glycolysis plus some like small reactions that regenerate NAD, which then can be reused by glycolysis to continue going through this until oxygen is back in the system. So there's 2 main types of fermentation, alcoholic and lactic acid. Alcoholic fermentation is when pyruvate is converted to ethanol and you release CO2. You've probably heard of this, maybe from the It's Time to Eat stations, but alcoholic fermentation is used by yeast, and it's used when you're like brewing beer or making wine or baking bread or anything along the lines where you use yeast to have something rise. By releasing the CO2, you end up with bread, muffins, donuts, anything baking that you use yeast with goes through alcoholic fermentation. Now the opposite, or not opposite, but the other type of fermentation is lactic acid. Pyruvate is reduced to NADH, which forms lactate as an end product. There's no CO2 being released here, but lactic acid fermentation is used by fungi and bacteria to make all your loved dairy products like cheese and yogurt. And it's also used in human muscle cells. So you know when you're like running or like going up a bajillion stairs and all of a sudden you have like a wicked cramp and you can't move and everything hurts? Yeah, that's because your muscle cells have ran out of oxygen and you're not breathing enough, right? You're taking like wicked deep breaths to try and get oxygen back into the system, and your cells have stopped going through cellular respiration and have started going through lactic acid fermentation. So what happens is, because you don't have oxygen, you've now built up a bunch of lactic acid, and that lactic acid leads to muscle cramps. Now, we did it. We're on our last slide. I know this was so long, but thank you for sticking with me. So this is just a basic comparison between the two. This would be like great to know if, you had a test on this. There might be a question about this. So just FYI. So both processes can start with glucose and go through glycolysis. They both produce pyruvate. It's what happens next that changes it. So if no oxygen is present, it creates ethanol or lactate. If oxygen is present, it will create acetyl-CoA and then go through the citric acid cycle, the Krebs cycle, to the etc., right? But the big difference here is cellular respiration produces a ton of ATP, whereas fermentation produces 2 ATP per glucose molecule. That's like chump change, right? That's like nothing. If you want to be making a lot of ATP and doing a lot of cellular work, you need to have oxygen present to be successful in that. Whew, okay, we did it. I am very proud of you for sticking with me, and good luck. We'll go over this a little bit more in class next time.